Hello everyone, welcome to Biotech Notebook. In this video, we are going to discuss last 5-year GATE biotechnology biochemistry questions. Okay, which of the following types of molecules act as biological catalyst? Protein, RNA, and phospholipid. As we know that enzymes are known as biological catalysts, and most of the enzymes are made up of proteins. Okay, there is one exception with that is ribozyme. Okay, so this ribozyme is made up of RNA. Okay, and this ribozyme also capable of catalyzing a chemical reaction. So protein can act as a biological catalyst, and RNA, that is ribozyme, can act as a biological catalyst. So the answer is P and Q only. Okay, so there is no enzymes made up of phospholipid. And the next question is, a protein without its prosthetic group is known as. A functional protein has two parts. One is protein part and another one is non-protein part. Protein part is known as apoprotein and non-protein component is known as prosthetic group. Okay, your protein without its prosthetic group is known as apoprotein. Okay, so the answer is apoprotein. So the option A is correct, apoprotein. Okay, next let's see what is hemoprotein, holoprotein and lipoprotein. So hemoprotein is nothing but a protein that contains heme. Okay, that contains heme as a prosthetic group. Next, holoprotein. So your holoprotein is nothing but apoprotein plus prosthetic group. So holoprotein is a conjugated protein, that is it has both protein component and non-protein component. So protein is made up of protein and fats. So the next question is, which of the following statements about reversible enzyme inhibitors are correct? This is your enzyme catalyzed reaction, and enzyme inhibitors are the substances which binds with the enzyme and it brings about a decrease in the catalytic activity of your enzyme. Here the question is, uncompetitive inhibitors bind only to the enzyme substrate complex. So in uncompetitive inhibition, your inhibitor can't bind with the enzyme itself. Okay, so only it can bind with the enzyme substrate complex. After substrate binding, it changes the inhibitor binding site. Okay, so this is your inhibitor binding site before your substrate bind to with the enzyme. So after your substrate comes and bind with the enzyme, there will be a conformational change in the inhibitor binding site. Okay, so inhibitor can only bind with the enzyme substrate complex in uncompetitive inhibition. So the first statement is correct. Okay, then the second statement is, non-competitive inhibitors bind only at a different site from the substrate. In non-competitive inhibition, inhibitor won't compete with the substrate. Okay, substrate will go and bind with the active site and your inhibitor will go and bind with the allosteric site. This is your general reaction for non-competitive inhibition. Okay, so here inhibitors can bind with enzyme or it can bind with enzyme substrate complex, because here your substrate has a site to bind with the enzyme, that is known as active site, and your inhibitor can go and bind with the enzyme and that site is known as allosteric site. So second statement also correct, your non-competitive inhibitor bind at a different site from the substrate. Okay, and third option is, competitive inhibitors bind to the same site as the substrate. Yes. In competitive inhibitor, your inhibitor resembles the substrate. Okay, so they both compete for this active site. So here inhibitor has more binding affinity for this active site. So inhibitor will go and bind at the active site and it will block the product formation. This is the equation for competitive inhibition. So here your inhibitor will go and bind with the enzyme at the active site. Okay, so all the options are correct. Next question, which of the following is all common features for both aerobic and anaerobic bacterial cultures? First, let's say what is aerobic and anaerobic bacteria. Okay, so aerobic bacteria need oxygen to survive. Okay, so that is known as aerobic bacteria, whereas anaerobic bacteria do not need oxygen to survive. Okay, in the question that they are asking, what are the common features of aerobic and anaerobic bacterial culture. So the options are glycolysis. Okay, so yes, glycolysis is common for both aerobic and anaerobic bacteria. So why? Because glycolysis takes place in the cytoplasm, then it is an oxygen independent process. Okay, so this process doesn't need oxygen, so this occur in both aerobic bacteria and anaerobic bacteria. So glucose is the source of energy. So in glycolysis process, this glucose is converted into 2 molecules of pyruvate. The next option is NAD plus is the oxidizing agent. Okay, yes, NAD plus is oxidizing agent for aerobic and anaerobic bacteria. Okay. Then third option is oxidative phosphorylation. So see here, stages of respiration, oxidative phosphorylation only takes place in aerobic bacteria. It does not take place in anaerobic bacteria. So this option is wrong. Okay, then they have given 2 net ATP molecules formed per glucose molecule. This is also wrong. Why? Because in aerobic bacteria, you will get 38 ATP molecules at the end of your process after these 3 stages. Okay, whereas in anaerobic bacteria, after these 2 stages, you will get only 2 ATP. So this is applicable only for anaerobic bacteria, not applicable for aerobic bacteria. So option A and B is correct. Next question is, which of the following plot is or correct for enzyme that obeys Michaelis-Menten kinetics? Assuming your substrate concentration is less than is equal to Michaelis-Menten constant. Substrate concentration, then Michaelis-Menten constant. Here V0 is the initial reaction velocity. As this concentration of substrate increases linearly, there will be increase in your initial reaction velocity. So your Michaelis-Menten equation is V0 is equal to Vmax into S divided by Km plus S. V0 is initial velocity, Vmax is maximum velocity. So your condition is your substrate concentration is less than Km. That is, compared to Km, your substrate concentration is very negligible. So you can write this equation as V0 is equal to Vmax into S divided by Km. From this equation, you know that V0 is directly proportional to substrate concentration. So at very low substrate concentration, this equation obeys first order kinetics. Your substrate concentration is linearly proportional to V0. So option A is correct and option D is also correct, because when time increases, your substrate concentration will decrease because your substrate is consumed by the reaction. It will convert it into product. So option A and D is correct. Next question is, which of the following are correct about protein structure? Okay, so first option P, secondary structure is formed by a repeating pattern of interactions among the polypeptide backbone atoms. Yes, it's correct. Second option Q, tertiary structure is a 3-dimensional arrangement of the polypeptide backbone atoms only. They are giving as like secondary structure. Here 3-dimensional arrangement of the polypeptide backbone atoms only. No, it's wrong. Secondary structure only formed by a repeating pattern of interactions among the polypeptide backbone atoms, whereas tertiary structure is formed by the 3-dimensional arrangement of an entire polypeptide chain. Okay, so option Q is wrong. Option P is correct. Then the third option are quaternary structure refers to an assembly of multiple polypeptide subunits. Yes, it's correct. There is a assembly of multiple polypeptide subunit. Option B is correct. P and R only. Next question is, which of the following chemical messengers or derivatives of tryptophan? Tryptophan is an amino acid which comes under one of the 3 aromatic amino acid, and it is the only amino acid which contains indole ring. Okay, then derivatives of tryptophan are used in the tryptophan metabolic pathway. Okay, so this question is comes under amino acid metabolism. To answer this question, you should know the end product, important intermediate products of your tryptophan metabolic pathway. Okay, so first one is gamma aminobutyric acid. Gamma aminobutyric acid is not a derivative of tryptophan. Then in tryptophan metabolic pathway, you have 3 types. One is kynurenine pathway, then serotonin pathway, then indole pathway. Okay, this indole acetic acid is produced in indole pathway. Melatonin and serotonin both are produced in serotonin pathway. In serotonin pathway, your melatonin is an end product and serotonin comes as an intermediate product. The answer is B, C, D. So indole acetic acid, melatonin, serotonin are derivatives of tryptophan. Okay. An enzyme E catalyzes a biochemical reaction. So your reaction is A gives B with K cat, that is catalytic constant. K cat is 500 second power minus 1. The initial velocity V naught, okay, V naught is 10 micromole second power minus 1 at the total enzyme concentration ET is 30 nanomolar. And substrate concentration, so your substrate is A, your product is B. So here they have given the substrate concentration A is 40 micromolar. They are asking what is the value of Km in micromolar, that is Michaelis-Menten constant. We can calculate Michaelis-Menten constant from the Michaelis-Menten equation. So your Michaelis-Menten equation is V naught is equal to V max into S divided by Km plus S. Here V naught is initial velocity, it's a given data. V max is the maximum velocity we have to find out from the given data, and S is substrate concentration, so it's given K, so you have to find out. So in this equation you have to find out V max and Km. So V max can be calculated by using this equation, that is V max is equal to K cat into ET. So K cat is given data, that is 500 second power minus 1, ET is total enzyme concentration 30 nano. Okay, so by substituting the given data in these two equation, you can find out the Michaelis-Menten constant Km. Okay, so V max is equal to K cat into ET. So K cat is 500 second power minus 1 and ET is 30 nanomolar. But Km they are asking in micromolar. So you have to convert 30 nanomolar into micromolar. So 30 nanomolar is nothing but 30 into 10 power minus 9 molar. You can write this as 30 into 10 power minus 3 into 10 power minus 6. So we know that 10 power minus 6 molar is equal to micromolar. So 30 into 10 power minus 3 micromolar. So Vmax is equal to 500 into 30 into 10 power minus 3 micromolar. 500 into 30 is equal to 15,000 into 10 power minus 3 micromolar. So these three zeros will cancel it out. So Vmax is equal to 15 micromolar. So we found Vmax. Now just calculate Km from this equation. So V0 we know that it is 10 and Vmax is 15 and substrate concentration given 40 micromolar. So Km plus 40. Just cross multiply. So 10 Km plus 10 into 40 400 is equal to 15 into 40 600. So 10 Km is equal to 600 minus 400 is equal to 200. So 10 Km is equal to 200. So Km is equal to 20 micromolar. Enzyme alpha amylase used in starch hydrolysis has an affinity constant Km value of 0.005 molar. So Km is equal to 0.005 molar. So to achieve 1/4 of the maximum rate of hydrolysis, the required starch concentration, they are asking the substrate concentration in millimolar to achieve 1/4 of the maximum rate of hydrolysis. That is, the velocity is 1/4 of your maximum velocity. Your Michaelis-Menten equation, V0 is equal to Vmax into S divided by Km plus S. V0 is your initial velocity, Vmax is maximum velocity, and S is substrate concentration. Km is your Michaelis-Menten constant. Here Km they have given is 0.005 molar and V0 they have given in terms of Vmax. V is equal to Vmax by 4. So Vmax into S we have to calculate Km 0.005 plus S. So Vmax will cancel it out. Then you just cross multiply. 4 into S is equal to, so here 1. So 0.005 plus S. 4S. So the substrate concentration will come to this side. So 4S minus S is equal to 0.005. So 4S minus S is equal to 3S is equal to 0.005. S is equal to 0.005 divided by 3. So that is equal to 0.00167 molar. So they are asking in millimolar. You can write this as 1.67 into 10 power minus 3 molar. So minus 3 molar is nothing but your millimolar. So substrate concentration is equal to 1.67 millimolar. The next question, protein concentration of a crude enzyme preparation was 10 mg per ml. So your protein concentration in your sample is 10 mg per ml. Okay, then 10 microliter of this sample gave an enzyme activity of 5 micromole per minute. You are taking 10 microliter from this preparation. So volume of your sample is 10 microliter. Then it gives an activity of 5 micromole per minute. So enzyme activity is equal to 5 micromole per minute, or you can write it as 5 units. So 1 unit is equal to 1 micromole per minute. Then the specific activity of this crude enzyme preparation is dash unit mg power minus 1. So they are asking specific activity of your enzyme in units per mg. Okay, formula to calculate specific activity is equal to enzyme activity in units divided by concentration of protein in mg. Enzyme activity they have given in units, 5 units, then concentration of protein in mg. Okay, so here they have given protein concentration in 10 mg per ml. We need protein concentration in mg. So to convert this 10 mg per ml into mg, you have to multiply your denominator of your formula with volume of sample. You can write this term as concentration of protein in mg per ml into volume of sample in ml. So here ml will cancel it out, then you will get concentration of protein in mg. First we have to change this volume of sample 10 microliter into ml. Okay, so we know that 1 ml is equal to 1000 ml. So 1 ml is equal to 1 by 1000, that is 10 power minus 3 ml. So 10 microliter is equal to 10 into 10 power minus 3. So that is equal to 10 power minus 2 ml. So just substitute in this formula. So enzyme activity given data, that is 5. Then concentration of protein in mg per ml is 10. And when volume of sample in ml is 10 power minus 2. So pi divided by this 0 will cancel it out. So you will get 10 power minus 1. So 5 divided by 0.1. Specific activity is equal to 5 divided by 0.1, that is equal to 50 u mg power minus 1. Which one of the following statement is correct for enzyme catalyzed reaction? Okay, the answer for this question is enzyme do not affect del G or KEQ. Okay, so the option D is correct, where del G is Gibbs free energy and Ke is equilibrium constant. Okay, let's see the reason. First, let's see what are enzymes. Okay, enzymes are biological catalyst. It increases the rate of biochemical reaction by decreasing the activation energy. Okay, so what is activation energy? Activation energy is the minimum energy that must be possessed by the reacting molecule, that is your substrate, before the reaction will occur for getting them into product. Okay, this is the activation energy required for the substrate for uncatalyzed reaction. Okay, if you are using enzyme in your reaction, so it will reduce the activation energy. Okay, so by decreasing the activation energy, it will increase the rate of the biochemical reaction. So this is the general function of your enzyme. Lg, that is your Gibbs free energy, is nothing but the free energy difference between your product and substrate. So look into the graph, the enzyme is changing only the activation energy. Uncatalyzed reaction and catalyzed reaction, there is no change in del G. Okay, so del G is same for both catalyzed and uncatalyzed reaction. So enzymes do not affect del G. Okay, so it won't change the energy level of your substrate or product. Then del G and K equilibrium can be used to predict the ratio of product to reactant at equilibrium for a given reaction. So the relationship is del G is equal to minus RT ln K. Okay, so in most of the chemical reaction, your substrate is in equilibrium with your product. Okay, so in general, enzyme increases the rate of the reaction by decreasing the activation energy. So it must accelerate both backward and forward reaction equally. So it won't change the equilibrium between the substrate and product. Then they do not affect del G and K equilibrium. Then these are the biochemistry reference book for GATE Biotechnology exam. So first one is Leninger Principles of Biochemistry, then Satyanarayana Biochemistry, then Palmer book for enzymes. Then you can also follow Biochemistry by White and White. Okay, so these are some of the reference book for biochemistry preparation. This is your biochemistry gate syllabus. So here you will have biomolecules, structure and function, biological membranes, structure, membrane channels and pumps, molecular motors, action potential and transport processes, then basic concepts, regulation and metabolism of carbohydrates, lipids, amino acids, nucleic acids, photosynthesis, respiration, electron transport chain, enzymes, also comes in biochemistry syllabus. So in enzyme part you have classification, catalytic and regulatory strategies, enzyme kinetics, that is Michaelism Menten equation. In every question paper you will have a problem which is related to Michaelis Menten equation. So you should know the problems which is related with Michaelis Menten equation. You can refer my solved problems enzyme playlist. Then enzyme inhibition, competitive, non-competitive and competitive inhibition. Okay, thank you for watching my videos.